So briefly, phages are bacteriophages are viruses that infect bacteria, and the infection often follows the same pathway that we see the infection of viruses infecting human cells. The bacteria attaches onto the cell. The bacteriophage attaches onto the cell of the bacteria, injects its DNA into the DNA or RNA, depending on what it is, into the cell of the bacteria, and of course, exploiting part of the mechanisms of the bacterial cells, it does cause either gets integrated depending on on the susceptibility of the bacteria, or is destroyed if the bacteria has evolved a restriction mechanism. Restriction mechanism. Just hold on. Joseph. My apologies, we were expecting a delivery, and unfortunately, the person delivering is just arriving now, and I had the connection to that. I'm sorry about that.
No worries,
Okay,
Misaki, no worries. Just go ahead. Yeah, just go ahead.
So essentially, when we see this happening, there is the process of attachment, entry, transcription, of course, of the viral DNA or RNA, and then, of course, the assembly of the bacteriophages within the bacterial cells. So what has happened is that the restriction modification systems have evolved as a primitive defense against phages. There is more or less an evolutionary arms race between the phage and, of course, the bacteria. The phage infecting the bacteria, which do not have RM systems, but the bacteria that then, of course, subsequently evolving or acquiring RM systems to defend themselves against the phages. So when bacteria acquire RM systems, they are protected against the infestation by these bacteriophages. So 2014 is this publication coming from PANAS. Restriction modification systems serve as innate immune defenses for bacteria. I thought this is a nice paper. Frankly, this has been known for years, from the time the restriction enzymes were identified. But how do they work? Restriction enzymes work by recognizing what we call palindromes or non-palindromic sites within the exogenous DNA, the DNA of the virus. So when they are deployed, the systems really exist as depending on the type of the restriction system. We have 4 types of systems, but they exist as either an independent restriction endonuclease enzyme existing along with the methyltransferase, the restriction endonuclease being responsible for slicing up the foreign exogenous DNA, while the restriction, the methyl endonuclease protects the host genome against attack by binding methylating sites within the host genome that are equally susceptible to the cleavage by the endonuclease. So the methylation literally is the process, and here this is exactly what I was trying to refer to, that when the host DNA is methylated, it is protected against cleavage by the restriction activity of the endonuclease. So on the side here, we see that there are about 4 types of restriction endonuclease systems. And I think we are realizing and coming to terms that even the CRISPR-Cas exist in different models. You have actually up to 4 classes of CRISPR-Cas. But you can see here the class 1 having 2 dimers of restriction endonuclease proteins and methylase against 1 specificity protein. The type 2 do not have a separate specificity protein. Both the restriction and methyl endonuclease have the activity of specificity. And of course, these solidarity DNA methyl transferases, DAM and DCM, are responsible for protecting the host genome. So in 1978, Nobel Prize was awarded to Aber, Nathans, and Smith for, of course, identifying these restriction enzymes in bacteria. This probably forms the seminal part of quite a lot of work that in terms of applications regarding molecular biology, but downstream, of course, evolution of some of the therapeutic applications that we are thinking through now. Of course, Albert discovered restriction enzymes, postulating that these enzymes bind to double-stranded DNA sites containing recurring structural elements. Smith verified the hypothesis with a purified bacterial restriction enzyme. And then Nathan's pioneered the application of restriction enzymes to genetics and demonstrated their use in the construction of genetic maps. And this is literally restriction fragment length polymorphisms and other things that we really play around with in molecular biology. So in terms of utility applications, restriction maps, gene optimization, organization, gene sequencing, we are using restriction enzymes, restriction fragment length polymorphism, forensic analysis, gene cloning, and DNA methyl sensitivity assays, and the nucleosomal DNA sensitivity assays. So here, what's happening is that these are the existing applications. Early in 2000, actually starting in 1999, we hypothesized that actually this model of bacterial restriction enzymes could form a model for a gene therapy against HIV. And my group, of course, doing quite a lot of work backwards in there between the year 2000 running through to 2000 and I think 10, identifying initially restriction endonucleases. Much of this work was done during my fellowship in Canada. That cut up the HIV and the different seed genomes. This work was published in the African Journal of Biotechnology. I must say this work was initially rejected by the big journals. We had sent it to the American Journal of Microbiology. And I remember one of the reviews was that, I mean, the paper contained sequences, I mean, things that could not be identified by the word document. Of course, a lot of DNA sites and other things would look irregular to DNA, to a word reader. So in 2005, a newspaper daily labeled The New Vision in Uganda carried a story after a presentation that we have made at a regional conference here. And this is perhaps the easiest way to explain this to a lay person. If you can access this article, it's published in February 28th, 2005. The daily is called New Vision. You should be able to access this article speaking about how we are planned to use bacterial-derived enzymes. Of course, in our mind, we we knew that there would be issues with the toxicity to the host genome, because restriction enzymes cleave at very short palindromic sequences, and it's very difficult to really ensure specificity for bacterial restriction enzymes, especially when you are applying them in the host as therapeutics. But we thought some of these could be applied by using similar defense mechanisms, including methylation of host targets areas. But we'll see how subsequently we've been able to move that. Of course, the biggest advance we had as far as the the development of this restriction enzymatic therapeutics is the evolution or emergence of artificial restriction endonuclease systems, particularly the zinc finger nuclease system, developed, of course, for many things, largely to help in terms of genome editing, and as well as, of course, host genome editing, but at the same time sequencing and other things. But of course, for us, we saw the opportunity, given the fact that we had come from backwards. We actually assembled a set of restriction enzymes in 2010, which cleave the HIV zinc finger, HIV pol gene. And our essence was the reason we wanted to target the pol gene is the pol gene codes for 3 particular enzymes involved in the reverse transcriptase. I think there is an integrase in there. I'm not sure about the 4th protein, I don't remember. But we thought that by targeting this gene and disrupting it, we would be able to abrogate the function of particularly latent HIV within latently infected cells. Of course, that's another issue that my colleague Keith will talk about, the concept of how do we target these cells, and those are some of the challenges we've been trying to overcome. But for us, we were looking at, number one, targeting a particular gene. We know that the host genome, for example, the human genome has residual retroviral elements which have been silenced historically. The silencing did not have to cleave away. There is the hypothesis of having to cut away excise the entire proviral DNA. And I think that is a very complicated thing. I think that the easier thing would be to go in, silence a gene, which is very important to the replication, and then subsequently this proviral genome would be disabled. In a similar way, we have the different restriction, retro elements that are integrated in the host genome. So a lot of work was done, of course, using different assays. The MEL assay is one of the assays that we use in vitro to demonstrate the functionality of restriction enzymes. And we assembled about 5 or so pairs of restriction endonucleases. But we decided to, of course, we had to pick up the restriction enzyme with the highest specificity in terms of targeting the polymerase gene. And then, of course, we assembled plasmids which were carrying the zinc finger map targeted towards the polymerase gene. And these plasmids were used to transfect vectors, including, of course, the adenovirus type 5. So we created, first of all, the plasmids themselves, were supposed to use them directly as a delivery vehicle in our experiments. We'll see that, how they perform, because they have been directly used for that purpose. But at the same time, of course, we are using the adenoviral vector type 5 as a delivery vehicle for transfecting human cells. These are the sequences for the CRISPR-Cas. We generated CRISPR-Cas9 targeting the polymerase gene as well. So in addition to the zinc fingers, we also worked with the CRISPR-Cas. And of course, the CRISPR-Cas have their own vector systems, and you will see how this works. The plasmid construct for containing the HIV gene specific CRISPR-Cas revolves around this. And you can see we have a CMV promoter running through this process. So in order for us to, of course, obtain in vitro proof of concept, we had to use cell lines, and we use different cell lines. For the active HIV infection, we use the TZM-BL cell line acquired from the NIH Research Reagent program. This TZM-BL cell is special in the sense that it contains a luciferase marker. It contains a luciferase gene, which is activated when the cell gets infected with HIV. That gene, its transcription is driven by the expression of the long terminal repeats for HIV. It's very interesting cell line, and sometimes when you sit down, you have to thank the people who developed these cell lines. I mean, who developed these cell models, because it makes it quite easy for us that are working on this other side. On the other hand, of course, we have different cells that are models for HIV latency, the SCH2 cell, and then the Ducat cell line, which also has a luciferase marker, a GFP marker within it. So briefly, we obtained the contract, and I think our contract ended last year. The AIDS reagent program has been undergoing changes. We need to renew this, but this was a 10-year contract to acquire cell lines. So we cultured these cell lines on growth media, of course, and then transfected them with either the zinc fingers or the CRISPR-Cas plasmids or the adenoviral vectors carrying the zinc fingers. The idea was to see whether we could reverse HIV latency. So in terms of analyzing latency, the large thing we are looking at is, first of all, the issue of there's the treatment of the cultures, but also the issue of the reversal of latency following the use of latency reversing reagents. These are HIV activating reagents, prostatin, phobo, mediastate, SAHA. A lot of work has been done around this as latent reversing reagents. These were, of course, obtained as a courtesy of GSK. Actually, this grant, a lot of this work was funded by the GSK Trust in Africa grant. So this is part of the molecular visualization in terms of the DNA ladders. I don't know. I think we will get to see how this works. Maybe these are initially, this resembles a male assay, but essentially when the DNA is being treated by the enzyme, you can either see whether it's being cut, the target is being cut, or it's not being cut. And this is the one way to prove. So this is ideally the cell assay that is being used to tell us whether the polyclonal is being targeted. But then what of off-target toxicity? Of course, we used tryptan blue to study the viability of some of the cells. And as we see, normally in cell culture, some cells die. So we really got to see some cells dying, but we don't think this was really driven by the treatment. Probably was an effect of what happens when you culture cells. So in terms of evaluating the other aspects, is it possible if you modify the human genome of people who are susceptible? Can we use this as a prophylaxis? Can we deploy these enzymes as a prophylaxis to protect people against incoming HIV virus. So to do this, we use the TZM-BL cell model for both in terms of demonstrating the transfection, but also demonstrating the protection against the HIV challenge. And briefly, using a TZM-BL culture plate, we prepared wet preparation for fluorescent microscopy and then went in for fluorescency. And one of the things that we clearly see is that in the negative control, the untreated cells, several hours after being exposed to HIV, those cells emit luciferase. But and this is consistent with the marker that is present in the TZM-BL cell. But the cells which were treated with the plasmids, the zinc finger plasmids, the adenoviral vectors, RAGD vectors carrying zinc finger nuclease or CRISPR-Cas were actually protected against HIV transmission. So I think I'm going to stop here. I think we have quite a lot to hear from Keith, and I don't want to take much of the time. A lot of people played a big role in doing some of this work. These are my students and a few of my colleagues at the university faculty. We have, of course, different funders, but different people providing reagents. And we just became a MAC Center of Excellence, by the way, for CRISPR-Cas, and that's how myself and Keith Jerome got to meet to speak at a MAC meeting on CRISPR-Cas. And so we are happy to be here and do the same thing at the Institute of Pasture. Thank you.
Misaki, thank you very much for this nice talk and nice result that you showed. So let me, before giving the mic to the audience, let me ask you, what do you think is missing now to go for clinic?
You know, very nice question, Mohammed, because initially, both zinc finger nucleases and CRISPR-Cas have gone to the clinic already. So the argument we've tried to make before to really go for phase 1, small phase 1 septic trials previously have not gone well with because we are looking at gene therapy. At the end of the day, gene therapy carries a lot of ethical connotations. And so we need to carry this through the animal models. So we need to be able to do this in the humanized mice, but also probably move to bigger animals like non-human primates. And this is the kind of work that Keith Jerome is going to be talking about. That's a lot of his area.
Absolutely. I have also another maybe naive question. So here you talked about HIV-1. So is
yes.
this the same maybe strategy for HIV-2, which I know it's a little bit minoritaire?
So frankly, the global distribution for HIV really is largely represented by HIV-1. So this prospect for the HIV-2, I don't think there are a lot of differences, but if you realize when we were identifying so many of the earlier enzymes, we try to look at the different types of either SHIV. SHIV is the simian HIV. This is for infecting, it infects non-human primates and causes HIV non-human primates. And tactfully, we did that, knowing that as we move to the animal models, the virus that we would be using for challenge would have to be a SHIV because it's what induces HIV in the non-human primates. So I think that the issue is not the design. The design can be modified for anything, literally. I mean, the technology is here, so if we choose to target, if we realize that there are differences, even in the subtypes that we have, for example, the pol gene we are targeting, then we should be able to come up with the enzymes that work towards a particular target.
Yeah. True. The audience, any question in the audience? I have also another. Yeah. Maybe you can come here to make it easier. Yeah. So waiting for the student, for the researcher to come to the. I have, because maybe you notice that I have some issue with Ebola. Can we do the same strategy for Ebola too, you think?
So frankly, two things. Ebola is a class A pathogen. And frankly, at this point, introducing any cleavages in Ebola is something that potentially we should not be thinking about for the sake of, unless we are going to do this with complete certainty that we will abolish the virus.
True.
But at the same time, it's easier to manipulate HIV in a BCL2 lab. So I do some work for developing diagnostics for Ebola, and one of our lead candidates is really moving fast because we're trying to, but we have to work with P4 facilities, and these are few within the setting of Africa. So it's very difficult work really to work around. So frankly, at this point, it would be something to think through. We need probably a moratorium to agree whether we should move this to high consequence pathogens.
Okay.
So, yes.
So thank you for your talk. So I have a naive question about your experimental plan. So at some points you say that you used both Zinger finger technology with CRISPR. What's the point about using both at the same time, and what's the difference between using only the CRISPR-Cas9 for HIV control rather than both at the same time? Thank you.
So at the time, first of all, to be clear, we never used both zinc fingers and CRISPR at the same time. But we compared the activity between the zinc fingers carried by just the zinc finger plasmids, and then compared it with the one carried, the zinc fingers carried by the adenoviral vectors, and then compared it with the CRISPR-Cas carried by the CRISPR-Cas plasmids. So we were not using all at a go. We were trying to compare between the activity. Why? Because at the time, I mean, CRISPR-Cas had just emerged. We wanted to see what is superior in terms of efficacy. We don't see a lot of superiority between CRISPR and zinc from the experiments that we did, but clearly what we see is that the adenoviral vectors increase the activity of the zinc fingers, bringing it closer to what we see in CRISPR. Of course, the other thing that is quite clear here is very difficult really to look at this. The zinc fingers have a lot of off-target non-specificity. Of course, that's really a question to think through, and Chris probably, Keith will talk about this. Is it that CRISPR really is more specific, or the older enzymes like the meganucleases can be designed to be more specific and probably will offer a better alternative? And these are things that I think the world is emerging through, because the biggest challenge for restriction endonuclease enzymatic therapeutics is really, or artificial restriction enzymes as therapeutics for use in viral or whatever, is really to ensure that we have specificity we ensure that this is specific for the target it's meant, and we don't have off-target effects coming from in the host genome. For example, for us, we are interested in targeting the virus rather than targeting. What I need to tell you, there are other groups that, for example, were working on the host pathway. And their idea was, for example, to mutate and abrogate or abolish the expression of, for example, CCR5. Because clearly, we know that mutations in CCR5, they are, including the Delta 35, are associated with protection against HIV. And actually, this has been demonstrated through the transplants that were done. I know you know about Timothy Ray Brown. It's the first person really cured from HIV, received a bone marrow transplant from Germany. They made sure that they receive the transplant. The donor was CCR5 Delta 32 mutant. And the idea was to see whether this could work. So there is a group working on the other side, imagining that they could do that. For us on the other side, our interest is to target the viral pathway. Why? Everybody has their reasons. The other group argues that naturally, people who are Delta 32 deficient or mutant, they are protected against HIV. For us on this side, we argue that the whole genome, human genome, is full of retroviruses, but they are not causing disease because they are being inactivated in one sense or another. The body has found ways of inactivating this retrovirus. So that's how we argue that perhaps we can do it this way. There are many challenges. Keith will talk about them in terms of really bringing this to the clinic.
Thank you. Thank you very much.
Thank you. So maybe any, yes, I mean. A fish question it will be, or
Fish
no?
one. Hi, sir. Thank you for your great job.
Thank you.
Actually, I have a question about the robustness of zinc finger and CRISPR to possible viral resistance or evasion mechanism. Have you assessed these possibilities?
So frankly, this is that's a very nice question, because the virus that we are dealing with is highly mutable as well. So it's partly explaining why we went for the polymerase gene, because by just targeting the polymerase gene, you are achieving more or less what combinational, highly active combinational therapy is doing. So you know historically what happened with the evolution of HIV treatment and retroviral treatment. Initially, we came up with few monotherapy drugs like zidovudine, and zidovudine, you would give it to a patient, it works very well in the first months, and then a new virus emerges, and then zidovudine is not working anymore. So frankly, the question you're asking is very important, and so brings us to the point why for us we chose the polymerase gene, because we believe that this gene codes for 3 enzymes, and by literally abrogating it, you are achieving an activity beyond just one enzymatic. But maybe we need to use a combo of enzymes targeting different CRISPR designs targeting maybe more than that. For example, in the HIV therapeutics, they have a nucleoside inhibitor with a non-nucleoside inhibitor, and then they combine it sometimes with either a protease or a reverse transcriptase. So maybe we can think about doing this. But that's a very nice question. Thank you.
Well, thank you very much, Misaki.